Please be sure you watch the DNA replication video before the protein synthesis video. This is the protein synthesis video. Just a little recap. DNA replication is your DNA having to make a copy because you need to produce more cells. This happens in the S phase of interphase as the cell is growing, and what ends up happening is the DNA strand is going to separate, and then it produces a new strand. Each one is going to produce a brand new strand, so you have two copies now of DNA. Now, what this lecture is about is how your DNA is going to actually produce proteins. If you recall, I think it was chapter 7, we said the main job of the cell's nucleus, which has the DNA, it's to contain the instructions for making proteins. Now, here's where we get to that. Here's where we find the instructions for making proteins. Okay, so now, while we talk about DNA and protein synthesis, we also have to talk about a new structure called RNA. So let's go back just a second. From chapter 10, we know that genes are coded DNA structure instructions, and they control all of the production of proteins in your body. They have to be decoded, and what's going to end up happening is you're going to take a part of your DNA and you're going to code that into a new structure called RNA. Now, this RNA is actually going to be what contains the coded information from the proteins. So what we're going to do is we're going to take the DNA, we're going to make an RNA strand, and then from there, we're going to produce proteins. I don't know if you guys are familiar with RNA, especially right now with the coronavirus, if you've watched much about that. The coronavirus actually isn't a DNA strand. It is a single RNA strand, and we're hopefully make you a little bit more familiar today about what RNA is. The way RNA is different from DNA, if you look at the picture, DNA is the two-stranded molecule on the right, and RNA is the single-stranded molecule on the left. Now, how they're different is it's called RNA because instead of deoxyribose that you have in DNA, RNA has the sugar ribose, so it's a ribonucleic acid instead of a deoxyribonucleic acid. Now, I just talked about RNA is generally a single-stranded molecule as opposed to the double-stranded molecule in DNA. And there is a difference with your base pairings. On RNA, it does not have the base thymine. It has uracil. It still has cytosine, guanine, adenine, but it does have uracil now. So whenever it is pairing up with the other side of the DNA, instead of adenine pairing with thymine, adenine is now going to pair with uracil. There are 3 different types of RNA, and they do actually have different structures. I don't know if we'll get into that today, but the first one that we'll deal with is called the messenger RNA, or mRNA. Then you have ribosomal RNA, rRNA, and then the last one is transfer RNA, or tRNA. When we talked about the structures of the cell, if you recall that the ribosomes are the actual site where proteins are made, and that is one reason the ribosomal RNA is important in this. Okay, so the first type you have is messenger RNA, or mRNA. And what this does is it is going to make a copy from the DNA. So messenger RNA carries copies of instructions for assembling amino acids into proteins. A really important part of this is many amino acids are put together and they are going to make the proteins. So the messenger RNA has all of the instructions, or it's going to carry the copies to the ribosomes. Now, the ribosomal RNA, the reason they're important is ribosomes are made up of proteins and ribosomal RNA, and ribosomes are what actually produce proteins. And the last one is transfer RNA, and what tRNA does is they are going to transfer the amino acids to the ribosomes for the proteins to be made. So let's talk a little bit about how this is going to take place. The blue top molecule is your DNA strand, which is the template, and then the red is going to be the mRNA that is made from the DNA strand. Now, so if I'm going from left to right, you have the nucleotide adenine and the red molecule U, which is uracil, because if you remember, instead of thymine, now you have uracil. Now moving on, you still have C pairs with G, and C pairs with G, and then you have another adenine, or A, and it has to pair with the U. And then, after you finish that entire strand, your mRNA, you're going to take those and make amino acids, and put all those amino acids together and make a protein. That was just a quick kind of overview of it. Now I'm going to go through it step by step. The first step that must take place is called transcription. Transcription is when the DNA is copied into that mRNA strand. So it's going to be when you're actually taking the instructions from DNA and producing the mRNA strand out of it. And still the same thing, that on this RNA strand there is no thymine, it has to be this uracil. Alright, so now let's talk about the genetic code. The genetic code is the language of the mRNA instructions. So your genetic code consists of 4 bases, or the 4 letters: adenine, uracil, cytosine, and guanine. The order of these bases are going to produce amino acids, and then many of those amino acids are put together, and then those produce proteins. Now when you put those bases together, you produce what is called a codon. A codon is 3 consecutive bases, or nucleotides, on the mRNA strand. And what those 3 codons are going to do is they are going to produce a specific or particular amino acid. Now, you don't have your books with you anymore, but in your books you have a codon table, and how you read this is that if I have the codon UCU, I know this is going to produce the amino acid serine. Okay, we all know that they have to have 3, so let's choose another one. If you have ACU, you have the amino acid threonine, and so on. So that takes us to translation. Translation is actually the decoding of the mRNA message. Translation does take place in the ribosomes, because this is where the amino acids are going to combine and form that protein. So what happens during translation is the cell uses the information from the mRNA strand that we just formed, and it produces these proteins. The ribosome binds the tRNA, the transfer RNA, and the amino acids, and then it's going to move along this mRNA strand. And there's kind of an example. Your tRNA strand is this. So it takes the codon, and then it is going to code, and then you're going to put all of those amino acids together and produce a protein. Some proteins are very long, and some proteins are very short. So they have many amino acids, or they can have fewer amino acids. It just depends. Now, if you look back at the codon table here, you see that you have different, you see it says stop. Your DNA strand is so long, you have to know when to stop that protein. And there is also start codons. And that's going to tell you, all right, here you're going to, this codon is going to start this protein, and once you get to any of these stop codons, you know that that amino acid is going to stop protein synthesis and your protein has been made. Now, stop codons are if they come into contact with UGA, UAG, or UAA. Alright, so just kind of a recap through this. So, at the top of this, you start out with a single-stranded DNA, and you are going to code it. C pairs with G, G pairs with C, T pairs with A, but over here, A pairs with U, because the mRNA strand does not have thymine, it has the uracil. This is our mRNA strand, and from there you're going to take each codon codes for an amino acid, and then put all these amino acids, and you get a protein. Many times they call it a polypeptide chain, just because these are held together by peptide bonds and that's where you get that. So a lot of times they call proteins polypeptides. I'm going to follow this link and kind of take you through a computer simulated version of producing a protein. And I will attach this PowerPoint, so if you would ever like to go on and practice with this, you can. Okay, so in this simulation, here is your DNA strand. It splits. Now we are left with half of the DNA strand, so one of the strands, and now I am going to go through transcription. And how I do that is if I have on my DNA strand an A, I'm going to produce my mRNA through transcription. So my A is going to pair with U. T is still going to pair with A. T pairs with A. Once again, A pairs with uracil since there's no thymine. C pairs with G. G pairs with C. A pairs with U. T still pairs with A. C with G. T with A. And so on. So I'm going to continue. So right now I'm going through what is called transcription. And I'm producing my new mRNA strand. There's my new RNA strand, or mRNA strand. Now from there, these are called codons. Now, what I have to do is I have to find my start codon so I know that it is going to start the protein. The start codon, the only one there is AUG. So I click on that. Now when you're reading this table, you go AUG, that is my first amino acid. Now this, what I'm doing here, is called translation. I am taking the codon and I'm finding the correct amino acid and I drag it over there. So there's my first amino acid, CUA, CUA is my second amino acid, GAC, GUG, valine, UUC, UAG. Now this says stop. I know that this is my stop codon and that is the end of my protein. So all of those amino acids put together are now a protein. So the first thing you do is make an mRNA strand, that's transcription. Then you're going to take the codons and find your amino acids, and that's translation. And that is the end of, now we have successfully created a protein.